As it is commonly known, different individuals react differently to food, plants, and medications. Therefore, compound may be served both as a drug and a poison at the same time. In this sense, a drug dosage that induces toxicity in a patient might also be able to treat another well, and at the same time had no effects on the other. Another important observations are the understanding of metabolic variability to unravel the mysteries behind these phenomena. This concept claims that biochemical processes within the organism are responsible for transformation of compounds, and that this process could be organic responses. Metabolizer subpopulation. There is now a general understanding that variability in the function of drug metabolizing enzyme is responsible for many differences in the disposition and clinical consequences of drugs. It is well known that drug levels can be raised by increasing the dose or by more frequent administration and vice versa. However, despite the advances in medical technology and potential predictive models, they are not yet sufficient to prevent high level of morbidity and mortality caused by adverse drug reaction. Therefore, it is believed that the study of how genetic variation interfaces with drug metabolism, especially in genes codifying DMEs, may also lead to improved drug safety. A variety of factors affecting the expression and activity of DMEs are classified into three major groups, which are genetic factors, non-genetic host factors, and environmental factors. Recent studies indicate that inter-individual variation in drug metabolism is one of the most important causes of drug response differences. In general, common pharmacokinetic profile is a lighthouse for most prescribers in clinical practice. Moving on to the next slide. Drug biotransformation by enzymes, represented by E1 in the diagram, has a key role in the control of plasmatic drug concentration. Low activity of the metabolic step might cause accumulation of the drug and its metabolite in the body. In this regard, genetic mutations in coding and non-coding regions may be involved in such inborn altered enzymatic activity. Some relevant examples come from polymorphism in cytochrome P450 genes, which may result in absence of protein synthesis, no enzyme activity, altered substrate specificity, reduced affinity for substrate, decreased stability, or even increased enzyme activity. Since the effect of inherited variation on enzymatic activity is the result of changes in DNA sequence, it is plausible that there are distinct subgroups of subjects who have different metabolic capabilities. Epidemiologic studies have revealed subpopulation of individuals based on drug metabolizing profile, which brings us to our next point, which is lessons from phase 1 and phase 2 reaction. The phase 1 metabolizing enzymes are responsible to convert drugs in reactive electrophilic metabolites by oxidation, hydrolysis, cyclization, reduction, and decyclization. The major and most common phase 1 enzyme involved are the macrosomal cytochrome P450 superfamily. A common target of study in pharmacogenomics is CYP2D6. Nowadays, it is well recognized that CYP2D6 polymorphism may result in 4 phenotypes according to enzyme activity, which are poor metabolizers, intermediate metabolizers, extensive metabolizers, and ultra-rapid metabolizers. Extensive metabolizers phenotype is considered as reference and is the most frequent in worldwide population. Poor metabolizers inherit two deficient CYP2D6 allele, which result in a significant slower CYP2D6 metabolism rate. Intermediate metabolizers are individuals carrying only one defective CYP2D6 allele. Since they still have some CYP2D6 metabolic activity, pharmacological responses in those patients are considered better than those observed in PM phenotype. And finally, the ultra-rapid phenotype, which result from gene duplication. The relevance of such genetic variation in the biotransformation of drugs is very impressive. First, at least 1/5 of all drugs used in clinical practice share a pathway in CYP2D6 route. Second, phenotype status directly affect the clinical response. It is worthwhile to mention some clinical consideration with regard to recent advances seen in pharmacogenomics. First, Although genotyping may be useful in predicting a drug response or toxicological risk, classical factors related with variability in drug response must also be considered at every stage. Second, it is widely accepted that genetic variability in DMEs are also directly correlated with susceptibility to unexpected outcomes, such as suicide, cancer, and other complex diseases. In other words, pharmacogenomic approaches are not limited to drug response.
Clinical pharmacogenetic and potential application. Primary cause of therapeutic inefficacy and pharmacologic toxicity is due to the individual variability in plasma drug level. For example, enzyme thiopurine methyltransferase, or known as TPMT, which this enzyme will metabolize two purine drugs, which is azathioprine, AZA, and its metabolite, the 6-mecaptopurine 6-MP. The polymorphism in TMP gene cause deficient in TMP activity. It will cause the thiopurine metabolism using different pathway and can lead into the bone marrow toxicity and myelosuppression. So, by identify the individual with low or absent of TPMT enzyme activity using the TPMT genotyping may provide useful tool for optimize the therapeutic response and prevent toxicity. Another example is NA2 gene. NA2 gene related to the isoniazide-induced hepatotoxicity. The metabolite responsible for isoniazide-induced hepatotoxicity was acetylhydrazine, ACHZ, which can undergo further metabolism by cytochrome P to toxic reactive acetyl-free radical in patient with slow NAT2 acetylation capacities. Next, about the ethnic characterization. Ethnic characterization is not sufficient enough to reach the pharmacogenetic goals. So what we'll do is that it will focus on the personal genomic. Both intra-ethnic or inter-ethnic variability for a specific allele in human population are extremely large and may have relevant clinical implication. For example, we have the NA2-SNP, which several NAT2-SNP will causing defective enzyme are heterogeneity distributed in the world, making 50% of Caucasian but only 10% of Japanese to have the NAT2-SNP. So the probability to predict the efficiency and accurately an individual response to the drug from a multi-ethnic study is very low. So in order to attempt to predict the genotypes and phenotype from ethnic perspective have become less meaningful. Basically, it mainly in parts of the world in which people from different regions have mixed intensively. So that's conclude that ethnicity not always serve as a smart point to define the drug regime because it's not emphasized about their biological component. Next is about the PGX approach. There's a question about the PGX approach, either it is cost effective or not. So basically the goal for the cost effective of the PGX technology is that we know PGX technology must be more practical than the available approach nowadays. So the data, but then again the data on the cost effectiveness of the PGX are currently limited. So that's why there's a lot of studies nowadays have shown that PGX is potentially cost effective but under several certain circumstances. But then the genetic variation that merely affect the drug elimination, which will always increase the frequency of an adverse effect or a common side effect related to the pharmacogenicity. And the cost effectiveness of the PGX also can be vary from the high to low depending on the illness and also the gene that involved. PGX also have all highlighted the needs to improve the quality of the evidence-based economic. It is possible that the rapidly decreased cost of the genetic analysis and knowledge of the therapeutic application of the PGX will always be able to be systematically to make PGX approach more and more cost-effective technology. And finally, about the ethics in PGX and also the final consideration. About the ethics, we know that the source of the PGX data is mainly from the human sample, and the consideration is that PGX nowadays is leading us to uncover several and potential application of the PGX in the clinical practice. So what pharmacogeneticians do is that they try to improve the health of people without any distinction